Okay, welcome to this taster talk, which is we recorded on the 31st of March, and this is an update on. I'm going to provide you with a little bit of background about the virology and the science behind the current COVID-19 outbreak. So I'm Dr. Sarah Gretton. I'm a virologist that teaches first year lectures here at the University of Leicester in the School of Biological Sciences. I'm going to give you a little bit of background behind the science of the current outbreak and some insights into how this links to our understanding of virology at the moment. So just start with a bit of a recap on what a virus is. So viruses can be described as obligate intracellular parasites. And what this means is they require a cellular environment to grow, and they essentially have a parasitic role. They take over cells when they grow within them. So what do viruses look like? What scale are we talking about here? Well, we've got a nice diagram just on the right-hand side of this slide just here. And at the top image here, you can see a 200 times magnification. And that's the kind of magnification you use typically to see something like a human hair. And what you can see is a series of small objects just at the bottom of that image just there. And if you magnify those again by another 10 times, then you actually see that this is a collection of a pollen particle and some different cells. So we have some lymphocytes, that's a white blood cell, some red blood cells, and a yeast cell. Now, in order to be able to visualize a virus, we actually need to magnify to a greater extent, and actually to be able to see a virus, we'd need to magnify around 20,000 times, and that would require an electron microscope. So viruses can't be visualised under a standard light microscope. But what we can see if we magnify to 20,000 times here is actually comparative sizes of viruses and bacteria. So bacteria are much, much larger than viruses, typically about 1 micrometer in size. So you can see, for example, here some Staphylococcus and some E. coli, but you can also see examples of some Ebola just here. So Ebola is one of the larger viruses, so that's 1.2 micrometers in size, or rhinovirus, which is a much smaller virus, which is typically 30 nanometers in size. So when we're talking about viruses, we're talking about things that are much, much smaller than our cells and even smaller than bacteria. So what's important to understand about viruses is they have to grow inside a cell. So in order to produce more viruses, they require a cellular environment, and those environments are specific to different types of viruses. So each virus will have its natural host, which will be a particular type of cell. Now, the reason that viruses require a cell to grow in is that they lack the genetic information and the cellular apparatus to either generate their own metabolic energy or to provide or to produce their own proteins. So they can't synthesize proteins or produce metabolic energy. And so what they essentially do is have genetic material where they can hijack a cell and the cell will do that for them. And similarly, they have different properties when it comes to multiplying. So usually for organisms, then they will grow and divide or have some sort of reproductive cycle. For viruses, they don't get bigger. Essentially, the way they reproduce is by within their host cell is producing a number of preformed components, which then are assembled and released by the virus. And so they don't have that classic process when you think about bacteria of growing larger and dividing and multiplying that way. They're these preformed units within cells. So as I tend to talk about COVID-19 and really just to sort of reiterate, so how does the COVID-19 outbreak fit into our standard understanding of virology? So what you can see here is just an image, and this is actually of SARS-CoV-1, but we think SARS-CoV-2, based on our current understanding, is very similar in properties. And like all viruses, SARS-CoV-2, which is the virus that's causing the current COVID outbreak, consists of two key elements, a protein shell and some nucleic acids, so some genetic material, and that essentially provides the information which will govern how the virus operates when it's in a cell. And some viruses also have what we refer to as a lipid membrane or an envelope, and SARS-CoV-2 is one of these viruses. And I'll talk you through the components of that just here. So what you can see here, just on the right-hand side of the screen, is, as I say, it's actually SARS-CoV-1, but our understanding is that SARS-CoV-2 looks very similar. And so what it's got is this genetic material is in the form of a positive sense RNA strand, and I'll talk a little bit more about that in a moment. But that RNA is encapsulated in what we refer to as a nuclear capsid protein, and you can see that just here. And then essentially it has a lipid membrane which contains a membrane glycoprotein. And then additionally to that, it has these spike proteins that protrude out of the surface of the virus particle. And that's quite important. That's actually the reason it's referred to as a coronavirus. So coronavirus, a large family of viruses which infect a variety of different organisms. And these coronaviruses take their name from the fact that they have these spikes on the outside, which give them a crown-like appearance, and so hence the Latin corona for crown gives them their name. And interestingly, you can see a little bit better just what they look like. So this is an electron micrograph, so an image taken on an electron microscope, and this is actually taken, just published a few days ago, but this is February publication, I should say. But this is just an image that's taken coronavirus from an infected patient and grown it in cells. And later imaged by electron microscope, and you can just see here the scale bar 100 nanometers. So coronaviruses are one of the larger virus families, but you can see here this typical crown-like structure which gives them their name. So I've talked a little about nomenclature here. So SARS-CoV-2 is the name of the virus itself, and that's essentially indicates SARS stands for Severe Acquired Respiratory Syndrome, and that's the disease that it causes. CoV is the coronavirus, and it's called number 2 because it's the second coronavirus in this group. SARS-CoV-1 is actually the virus which caused the first SARS outbreak in the early 2000s. And there's some interesting work to be done on how SARS-CoV-1 and SARS-CoV-2 compare. So actually what we think at the moment, and I'll talk a little bit more how this fits into the virus life cycle, but some research that was just actually published in the last couple of days shows that these spike proteins on the outside of SARS-CoV-2 are able to bind with a much higher affinity to the receptor that allows the virus to enter a particular cell. And so we think this is why SARS-CoV-2 has some of the properties it has compared to SARS-CoV-1, is it's able to bind to these cellular receptors with a much greater affinity than its close relative, and that results in some of the differences in, I suppose, pathogenicity and disease prognosis that we've seen with SARS-CoV-1. So one of the key differences that's noticed early on between this outbreak and the previous outbreak has been that SARS-CoV-2, people are infectious before they develop symptoms, and that's one of the reasons why this outbreak is much more severe. So if you remember, the first SARS outbreak was mainly confined to China, and the reason that they were able to confine the disease was because typically people showed symptoms 24 to 36 hours before they became infectious. Now, unfortunately, the converse is true, and our current understanding is with SARS-CoV-2, and that people can shed virus before they develop symptoms, and this has meant it's been much, much harder to track and trace people that have the virus, and therefore we have this situation where potentially people can be infecting other people before they realise they're ill. Okay, so where does the virus come from? Well, this is a nice image here, again from a recent publication. This has just come out in the last month or so from Nature, and basically shows the relationship between different viruses within the coronavirus family. And so how scientists really gather this data is to actually sequence the genetic material of the virus and then compare it to other known sequences. And essentially, the closer the sequence is to another one, then we can assume the more closely related those different viruses are. Now, what happens with viruses, and particularly with this virus, because of its genetic structure is that they're constantly mutating. But what that allows us to do is to compare those viruses that are closer, and we assume that those that essentially the viruses that have similar sequences are ones that have had a small amount of mutation and are actually, as I say, quite closely related. So what we've got here is both the alpha coronaviruses and the beta coronaviruses, and what you can see just in red is what we refer to as SARS-CoV-2. So when this paper was published, it was actually called the 2019 coronavirus. And what you can see is they've actually taken samples from 5 different patients, all infected or treated in Wuhan, and they basically sequenced the RNA of those viruses, and what they've done is compared it with known viruses that are already existing in a sequencing bank. And what we can see here is that the particular virus we're interested in, the SARS-CoV-2, is very closely related to a coronavirus that's been circulating in the bat population. And so when we think, where does the virus comes from, like many of the infections that we've seen on a global basis that have been particularly concerning, is that essentially it comes from an animal population. And what we don't know exactly is what properties allow the virus to cross the species barrier, but essentially if the virus is able to enter a cell based on it being able to dock and bind to a receptor effectively, then that will allow it to infect different species. So it's all dependent on the I suppose the properties and the protein structure of those spike proteins and how well they're able to bind to receptors, whether that be in an animal cell or in a human cell. And what we can see here is that this seems to be very closely related to, as I say, to a bat virus. And so it seems likely that there's been some minor mutations in the bat virus which have resulted in the virus that we're now seeing infecting and causing this current global pandemic. What we don't know for certain is exactly how this entered or how humans came across this. And one of the current theories is that actually it didn't come directly from bats, but there was possibly another species that essentially was a vector, which allowed essentially contracted the virus from the bat population and then transmitted it to humans. And it's thought that this happened in one of these live animal markets in China. And that's one of the reasons we have to think carefully about close contact with animals, is that close contact between humans and wild animals is a really excellent way of transmitting viruses from an animal population to a human population, the technical term for that being zoonosis. So how does this virus actually work when it enters the cell? How does it carry out its function? So viruses, as I mentioned already, are only able to enter specific cells. And if you look here, this is just a nice image of the coronavirus. Here you can see the classic spikes. And this is actually a paper which reviews the family of coronaviruses and how they enter cells. And our understanding of SARS-CoV-2 is it operates very similar to SARS-CoV-1 and MERS, which is the Middle Eastern respiratory virus, which is also a coronavirus. So this paper here shows how the classic structure of coronavirus with these spikes allow it to engage with a receptor. And as I mentioned, the receptor for SARS-CoV-1 and SARS-CoV-2 is an ACE2 receptor, which is found in the upper respiratory tract. So that will allow it to specifically enter cells of that part of the body. And what we think happens here is that the virus binds and that's with a receptor, and that allows essentially it to be assimilated into the cell. Essentially it provides a kind of unlocking mechanism in which the cell envelops the whole virus particle within the cell. Once this happens, the virus is unpackaged, and the important piece here is essentially it releases the genetic material. And the genetic material can then take over the function, or allows the function of the virus to take over the cell. As I mentioned before, SARS-CoV-2 is what we describe as a positive sense RNA virus. And what this means is it has an RNA in the positive sense, and this would be analogous to the messenger RNA that a cell would naturally produce when it was undergoing going through the process of transcription and translation. So if you remember transcription and translation, then during that process, the DNA in the nucleus is transcribed into a messenger RNA, which is a positive sense orientation, which then exits the nucleus, moves to the endoplasmic reticulum, where ribosomes then translate it into protein. Now, the positive sense RNA of a coronavirus works in exactly the same way, so it can immediately be read in the cytoplasm of the cell by some ribosomes, and you can see the ribosomes just here on this image, and it actually produces two proteins. So it has two open reading frames in its RNA genome, and it produces these two proteins from those open reading frames, and they get further broken down by a process of proteolysis into a set of proteins, which are then form what we call the replicase transcriptase complex. And essentially, these proteins don't form part of the virus structure, but what they do is provide the appropriate environment in the cellular machinery for new virus genetic material to be synthesized. So essentially, they form a structure together, and they actually form what we refer to as double membrane vesicles, and we think these are budded from the endoplasmic reticulum. And what they're therefore able to do essentially is to produce essentially a negative sense copy of the original, so complementary to the original positive strand RNA, which allows them to keep on producing more positive strand RNAs, which can go into the new virus. And so there's quite a complex process of transcription, but at the same time, so that transcription will allow us to produce the genetic material for the new virus particles, but at the same time, we also have a process of translation. And so one of the things that's also translated from these from this RNA is used to also produce more of the viral proteins which will form part of the new viral particle. And so these are translated at the endoplasmic reticulum, and they actually combine with something called the ergic, which is the intermediary between the endoplasmic reticulum and the Golgi apparatus. So essentially, the nucleocapsid proteins combine with these membranes, and that allows them to essentially assemble together using hijacking part of the standard pathways within the cell. They essentially are encapsulated with a smooth wall vesicle, and eventually they become released. And so the lipid in the virus that we see as part of this lipid envelope of the virus actually comes from taking part of the lipid away from this Golgi intermediary complex. So essentially the virus is released, and we sort of have typically two types of viral release, those which are released lytically, so essentially the virus breaks open the cell, or viruses like this, which actually hijack the endocytic pathway, and so the release of the virus here, the virus is essentially secreted out of the cell and don't actually result in death or lysis of the cell itself. So what actually happens? So this is the virus life cycle. It takes over the cell and essentially kind of quietly gets on with its business of producing both new genetic material and proteins that either support the production of new virus particles or become incorporated in the virus itself. But why does this actually cause disease? And it's interesting. I'm not going to talk in huge detail about this because this would be several lectures, which you can come and enjoy once you take your place at the university. But the immune response essentially is one of the key indicators of this virus as to how the likely prognosis and outcome for someone that is infected by SARS-CoV-2. And one of the things you might have come across in your reading around the virus is that some people develop severe respiratory failure, and we think that this is due to actually essentially what we would call an immunomodulatory or a modulation of the immune system, and essentially an overreaction to the virus. And this actually results in something called a cytokine storm. Now, cytokine storms can happen in a few different contexts and fashions, but essentially what happens here is that we have, as I say, an over-response to the virus. This results in a number of different white blood cells, so immune cells that you can see just here. And the example I've taken here is actually of what happens in influenza, but our understanding is what happens in SARS-CoV-2 is very similar. But essentially, this results in an overcompensation and actually migration of these immune cells out of their standard environment and into for example, the blood vessels itself. And this is what results in a malfunction of the lungs and essentially a difficulty in the body actually acquiring oxygen from the lungs and then transmitting it to the cells within the body. And it's this cytokine storm, we think, that's responsible, as I say, for the difficulties in breathing and therefore the requirement of people to go onto ventilators. So how does this map with what we're doing here at the University of Leicester? Well, we're actually doing some really fascinating research looking at how we can develop new tests which are both simple and efficient to understand the spread of the virus. And we've got some really nice articles and information on that. So if you want to go to the main university website, then you can read a little bit more about that. Thank you very much for your time and attention. And look forward to welcoming you to the University of Leicester later on this year.